Okay, this is the class questions for component 2, and this is question 1, the lovely calculate indexes of biodiversity question. So, obviously they don't give you any information that you don't need, so it's important to read the question. So, we're investigating the effect of pollution on species diversity in a stream. Sampling an unpolluted stream and a polluted stream, and recorded the number of individuals of different species present. So our first thing to do is to identify the dependent and the independent variables. So quite a few of you, when you did this, put dependent, oh, that's what we measure. So what do they tell us that we've measured? We've recorded, so this is the number of individuals of different species. And I know there's only a little short dotted line. Don't take shortcuts. Use the dependent variable that they've given you. So, the number of individuals of each species. And the independent variable, we've sampled an unpolluted and a polluted stream. So the independent variable is the pollution level of the stream. That was quite straightforward. AO1. Identify one possible source of error. So this is where you really needed to do a little bit of thinking here. So when we're doing animals, the problem with animals is that they move about. So you might not be able to catch them all in your area of sampling. You might count them twice if you're just kind of, pointing at them. So you've got animals move. They might have misidentified them, so misidentification is always an issue. And here we go, calculation. So we need a calculator. And we've got the results for the unpolluted stream. And we need to, so what have we got? We've got N, big N is the total number of animals for everything. We've got number of individuals per species, and we tell you that that means sum of, it means add them all up. So we need to add up these, and that will give us big N. So here we go. 4 plus 30 plus 70 plus 34 plus 10 plus 2 equals 150. So N, that's 150, times 150 minus 1 times 149 is 2, 2, 3, 5, 0. Then we're going to do the sum of N minus 1, which they've helpfully filled in for us. So really in an exam they don't want you to spend too much time punching numbers into a calculator. Pretty much what's important is what you do with it, how you interpret those numbers, as we shall see. That's 6926. And then we're using the formula to calculate the diversity index. Give your answer to 2 decimal places. So that's really important. Often at the end of a calculation they kind of tell you what to do with it. So 1 minus the sum of N minus 1, so that's 6926, divided by 22350. So on my calculator I've still got the number 6926. I'm going to divide that by 22350. So 1 minus 0.31. So all I've done is round up that 9 to 0.31, because if I'm giving my answer to 2 decimal places, there's no reason not to round. I'm then just going to use my calculator to stick a minus sign in front of it and add on 1. It's 0.69. Excellent. So you'll see that on your papers when you get them back, probably if you've written 0.69 on this page, somebody will put 3 ticks next to it. And if you've not got 0.69 on that page, they might have gone back and ticked that you can add up the numbers correctly. Rather appallingly, some of you didn't manage to add up those numbers correctly and do this calculation here. That was a bit bad. So, diversity index of the polluted stream was 0.1. What can you conclude from this figure? So this is a bit of an AO2, isn't it? So, you know, making a conclusion, analysing some data, what you do with the numbers. So what can we conclude from that? The polluted stream's 0.1. So we're not going to talk about the diversity indices being different, because we've been told the diversity. You've calculated one, you've been told the other. So we want to say something else. So we can talk about biodiversity. So the polluted stream has less biodiversity. Or you could say it has less individuals or types of species. The number of species will be different. Okay, a bit more AO2 here. So one student calculated the diversity index to be 10.24. What can be concluded from this result? So what do we know about diversity indices? They pretty much, because you're doing 1 minus, they pretty much go from 0, nothing there, to 1, lots and lots of things there. So can't be 10.24, that's too big a number. So what can I conclude from that? They've done something wrong. Now probably they've not done anything wrong with their sampling. So if you did, you know, miscalculated from those numbers, you're going to get a different number from 0.69. And probably what they've done is the calculation the other way round. They've put the n, this one, on the top perhaps. However, you're not expected to work out actually what mathematical error they've made. So what I can conclude is the student has made a mathematical error. Not an error in sampling, mathematical error. As the index of diversity is between 0 and 1. It can't be higher than 1. So, turning over. Ooh, the banded snail, one of my favourites. Do like a bit of Sepia nemoralis. Don't know how you pronounce that. Sepia maybe. So, we've got quite a lot of information here, so I'm going to read all the information first before I start on the questions. So, banded snail, found in a variety of habitats. So we've got grassland, heathland, sand dunes, bases of hedges, and the shell colour can vary. And then, presumably in an exam, this would be a lovely colour picture. So we've got yellow, brown, pink, white. The shell can be unbanded or have up to 5 bands. So we've got 2 characteristics here, and we've got 4 different colours, and then we've got unbanded, 1 band, 2 band, 3 band, 4 band, 5 band, and 6 banding patterns. So the morphologically different snails are all the same species. Explain. So let's give a reason why it's an example of genetic polymorphism. So what do we know about genetic polymorphism? I'm pretty much thinking about different alleles and heterozygosity. So, students made the following observations over a period of time. Predator is the thrush. You get fewer yellow eaten in summer, but the opposite for banded snails. In habitats such as tangled dense hedges, dark banded snails are not eaten. So, why? Why is that? And few yellow snails are eaten from grassland, but banded are. Again, why? Why is that? Explain how genetic polymorphism, so explain, give a reason, is maintained. So why have we still got all of these different bands and colours? Because, yes, so you need to be thinking about that while you answer the top bit of the question. So, morphologically different snails are all the same species. Explain why it's an example of genetic polymorphism. So, the different colours and banding patterns are caused by different alleles. Not by mutation alone. So if it was sort of, just down to a sort of quite a high mutation rate, you wouldn't get all of that huge variety. So what are we going to say about how it's maintained? Why we've still got yellow and banded all over the place? So I think the first thing we can see is the dark banded snails in habitats like dark dense hedges are not getting eaten, but the yellow snails are, and yellow snails are eaten from grassland but banded, so that are not eaten in grassland but banded are. So there's got to be a camouflage thing going on. So snails that are camouflaged in their habitat are not eaten by thrushes. And if we're going to put in a lovely, they have a selective advantage. That's a lovely word to use when you're talking about natural selection. In that habitat. Now you think, oh well, that would mean that you're only going to get dark banded snails in the hedges, but in summer the yellow banded snails, yellow shell snails, survive more. They are not eaten. There's probably a huge variety of ways to put that. And then this means that at different times of the year and in different habitats males survive. So that's going to maintain that sort of all that gene mixing and genetic polymorphism indeed. Okay, enjoy.